Evolution stands as one of nature's most remarkable and powerful forces. Through countless random mutations across the genome, paired with the persistent selection of variants that best adapt to their environment, life on Earth has flourished in unimaginable ways over millions of years. This force can be used in science to evolve proteins with desired functions and then unlock issues in a wide range of fields. Better proteins, directed evolution, new technologies, plastic pollution, sustainable development, artificial intelligence. We are the Evry Paris-Saclay 2024 team, and we're going to introduce you to our project PHAGEVO, a novel tool whose objective is to advance protein engineering and research.
PHAGEVO is a novel tool that combines phage-assisted non-continuous evolution, also known as PANCE, on the evolution T7 system, providing a powerful method for in vivo protein evolution. As a proof of concept, the project focused on evolving the XylS transcription factor to detect plastic degradation product, Phthalic Acid and Terephthalic Acid. Our project also leverages the power of artificial intelligence to engineer XylS for PA and TPA sensitivity owing to Flint, a model developed by our dry lab team based on PAGEVO. Dry lab and wet lab convergently managed to identify new XylS variant with improved sensing of PA and TPA, proving the interest and efficiency of PHAGEVO on Flint. We will now present you how we managed to reach this goal and why this work is important for the broader community.
Protein engineering involves mutating DNA sequences to generate protein mutants with optimized or novel properties, such as stability, ligand specificity, and enzymatic efficiency. There are three main strategies for protein engineering: rational design, semi-rational approaches, and directed evolution. Unlike the first one, directed evolution had the advantage of not requiring in-depth knowledge of the protein 3D structure or mechanism, by introducing random mutations into the genome. In our project, we aim to develop a cutting-edge protein engineering tool based on this approach.
Let's talk about the methodology of our project. So, PHAGEVO is the combination of two former technologies, PANCE and Evolution T7. While PANCE enables efficient selection of functional variants, actually it lacks of specificity, genes targeting. So we decided to combine with Evolution T7, which is a system that was developed by Evry Paris-Saclay iGEM team in 2021, which enables targeting and modularizing double-strand in vivo mutagenesis using RNA polymerase-guided deaminases that introduce mutation in specific gene sequences. Here, PANCE rely on protein 3 or 6 of the bacterial phage M13 for bacterial infection as a selection system. This phage is a circular, positive, single-stranded filamentous phage, whose infectivity is depending on protein 3, which specifically recognizes E. coli, F. pylori to enter into the cell. When in the absence of P6, phage particles remain associated with the host cell membrane. Our PHAGEVO technologies leverage this feature by making P6 selection marker for phage production, depending on the activity of XylS, which is here a gene of interest. In PANCE, the phage genome is depleted from the gene coding for P3 and P6, which is replaced by the gene to evolve. The missing gene is provided in trans expression from another plasmid. To evolve the gene of interest into a desired function, P3 and P6 expression level must be depending on fitness of the gene of interest for the desired function. Deleterious mutation will be eliminated as P3 and P6 won't be produce any phage, and the phage will not be infections, while beneficial mutation will produce phage amplifying the best of the gene of interest variants.
However, mutagenesis in PANCE is not targeted on the gene of interest. It occurs everywhere in the phage, plasmid, and bacterial chromosome. This can lead to deleterious mutations and loss of interesting one, thus lowering efficiency of the directed evolution experiment. This is what PHAGEVO wants to cure. We inserted in the phage genome the evolution T7 cassette flanking the gene of interest. It is based on the orthogonal T7 RNA polymerase linked to cytosine and adenine deaminase. Once modified RNA polymerase is expressed, the sequence flanked by T7 promoter rate terminator gets mutating with C to T or A to J transition mutation. Evolution T7 introduced also T to C and J to A mutations owing to a second T7 RNA polymerase working in the opposite direction. It leads to improved mutation rates compared to previous technologies based on polymerase-linked deaminase. We also created the E. coli S2060 delta Young delta NFI strain, specifically designed for PHAGEVO. The S2060 is a strain developed for phage-assisted continuous evolution derived from the well-known DH10B strain, with mutation-relieving issues in continuous culture, enabling robust induction of mutagenesis or evolution of specific gene of interest. On the contrary, E. coli MJ1655 delta flu delta PRF delta Young delta NFI is a strain developed to increase the mutation rate in base deaminase T7 RNA polymerase-based mutagenesis tools. Combining the specificities of these two strains was a prerequisite for successful evolution with the PHAGEVO system.
In today's age of biology, it's not just about trial and error anymore. We use AI to predict how mutations affect a protein's ability to interact with its target. Let's talk about PHAGEVO. PHAGEVO is a conditional generative model, which means that it imagines new protein sequences with a purpose to improve interaction with the target molecule, such as binding affinity to a ligand. This process involves intensive math, and while we won't detail the equations here, they will be displayed for those interested. Our work, Flint, enhanced interface for PHAGEVO simplifies the process. Run PHAGEVO with one terminal command, and it provides summaries of the generated mutants. How does PHAGEVO work? The binding pocket and ligand are represented as 3D point clouds, allowing PHAGEVO to better represent their interactions. PHAGEVO focuses on modifying the binding pocket, which directly interacts with the ligand, while keeping the rest of the protein structure fixed. The protein is modeled as a geometric graph of atoms, with nodes representing atomic groups and edges their spatial relationships. To capture the relationships between atoms and residues, PHAGEVO uses an attention mechanism, which identifies what's important to focus on during optimization. This mechanism works on two levels: atom-level attention, in which the model considers how well each atom in the pocket interacts with the ligand, and residue-level attention. On a larger scale, the model looks at entire residues and their relationships. Once the binding pocket is generated, PHAGEVO refines it using protein language models, in our case iGEM, which leverage evolutionary data to ensure the generated sequence is biologically feasible. Protein language models like AlphaFold treat protein sequences like sentences, learning patterns from vast datasets to predict protein behavior and structure. Throughout training, PHAGEVO uses a loss function to measure how well it is performing based on certain criteria. Specifically, the loss components predict the types of residues in the pocket and ensure that the model maintains the correct spatial arrangement for effective binding. In summary, PHAGEVO combines machine learning, evolutionary biology, and chemistry to create optimized protein binding pockets. Flint, our new interface for PHAGEVO, streamlines this process, making the tool more accessible and easier to use for researchers and developers alike.
As a proof of concept, we are using our tools to evolve a transcription factor found in Pseudomonas protida, XylS. This protein has been extensively studied and engineered for mutants with improved specificity or sensitivity toward one target molecule in order to serve as an efficient biosensor. Initially sensitive to a vast array of benzoic acid derivatives, specifically for mtoluic acid, we aim to switch its specificity, develop a stronger affinity for plastic degradation by products like Phthalic Acid, PA, and Terephthalic Acid, TPA. These compounds are natural ligands, making it useful for biosensing environmental pollutants and evolve enzymes with increased plastic degradation capabilities. Finally, to confirm the success of evolved XylS variants, we are using a bacterial reporter system which lights up when XylS binds to PA or TPA. This real-time readout lets us track how well the evolved proteins are working.
According to GFP fluorescence measurement, wild-type transcription factor XylX isn't sensitive to PA and TPA, in contrast with mtoluic acid, benzoic acid, and trichlorobenzoic acid. Two XylX mutants described in literature show slightly improved activity upon PA induction, but not with TPA. Starting from them, we performed directed evolution with PHAGEVO. Out of 30 parallel evolution experiments, and after a two-step screening process of nearly 3,000 clones, 5 interesting clones were revealed, all with induction significantly improved compared to wild-type and mutants already described. 2 of them reach induction ratio of respectively 4 and 2.4 at 1 millimolar PA. 2 others have induction ratio surpassing 1.4 at TPA concentrations as low as 100 micromolar, and PHAGEVO DE20C3 goes over this limit at 500 micromolar TPA. From the dry lab, 13 variants were generated with Flint. One of them has higher specificity and sensitivity to phthalic acid with an induction ratio reaching 2.5 at high PA concentration, and no measurable sensitivity to M-toluate, a feature never described in literature and a considerable improvement for biosensing purposes.
PHAGEVO is an ambitious and innovative project aiming to revolutionize protein engineering through directed evolution and artificial intelligence. But the impact of PHAGEVO extends beyond the lab. Ethical considerations and societal needs are central to our work, and the whole team was committed to the respect of best human practices. This project was driven by one goal: contribute to the broader scientific community by offering a resource that can catalyze innovation and address complex challenges in various fields, from environmental sustainability to healthcare. Our project's commitment to combating plastic pollution highlights the importance of addressing environmental responsibility, plastic waste being one of the most pervasive pollutants on the planet. Engaging in meaningful conversations with researchers helped us reflect on the broader implications of our work, ensuring we stay responsible and thoughtful in our approach. At PHAGEVO, we believe that clear, honest communication is key to overcoming fear and skepticism from the public. We have actively engaged with a diverse range of audiences through public talks here in Paris, but also abroad in Lebanon and Tunisia.
To address concerns around synthetic biology and directed evolution, we designed PHAGEVO with safety as a top priority. Our approach includes strict containment protocols to prevent the evolved sequence from escaping into the environment, and mutation targeted solely on the gene of interest, avoiding unintended changes in the E. coli genome. Using established risk assessment methods, we also created a short questionnaire to evaluate potential risk of directed evolution experiments, helping ensure proper safety and containment measures are in place from the start.
The PHAGEVO team will keep working thoroughly on the project to better characterize the mutants generated by PHAGEVO and Flint and identify the best XylS variants for PA and TPA sensing. Our subsequent goal is to feed the artificial intelligence model with data from the wet lab to combine the strengths of artificial intelligence and directed evolution, and propose new XylS variants with further improved properties. Additional developments of the Flint model will aim to deal with allosteric effects by incorporating them into the loss function. We also identify possible improvements for PHAGEVO that can be implemented to further increase its potential and make it a more powerful tool for use in synthetic biology laboratories around the world. Looking ahead, we see PHAGEVO at the forefront of protein evolution, with application in environmental sensors, industrial enzymes, pharmacological and medical fields, demonstrating the transformative power of synthetic biology. PHAGEVO's success in protein evolution contributes to the development of pollution detection technologies, helping to create a cleaner and more sustainable future.
This project emerged from the collaboration between our diverse and skilled wet lab and dry lab team members, whose expertise in cell and synthetic biology, structural biochemistry, bioinformatics, and programming created a true symbiosis. CEA Genoscope gave us access to their laboratories and excellent equipment, allowing us to develop our project to the best. By working together, we integrated our wet lab and dry lab efforts in the creation of this project. It could never happen without the supervision of our PI, Dr. Ioana Popescu, and the precious support of our 2 advisors that are PhD students at the CEA Genoscope. We are also deeply grateful for the support of our sponsors. Thank you. Synthetically and iGEMly yours,